Okay, hi everyone. I think we're about ready to get started. So I'll just give you a quick introduction. First, thank you all so much for coming. We are really excited to have you. Our speaker today is Susan, who will be going through the foundations for VCE biology. Just a little quick information about us. We are KIS Academics, and we're hosting this to help you get that head start and make sure you're starting year 12 on the right foot. If you have any questions throughout the talk today, we'll have some question time, so please put them in the Q&A function. We also might be doing some multiple choice polls, so make sure you get involved with those when they're up. And last but not least, hopefully you received the booklet from me this morning. Please have that with you so that we can pinpoint anything that you want to take note of, and so that you can annotate if that will help you with your learning. This recording will also be available on our online courses function on our website for KIS. So if you find this super helpful and you think your friends would also find it super helpful, they can watch it back after going through the paywall and everything. So yeah, instead of live tickets, it'll be an online course, which is super exciting as well. So thank you all for being here today. And without any further ado, I'll hand over. So Susan, when you're ready.
Okay, hello everyone. So I'll be covering the Back2Basics biology. So just a quick introduction. I am a second-year undergraduate med student. I received a 98.3 ATAR and I graduated in 2020 as the high school docs, and I received a 44 raw in biology. And there's just some pictures from my year 12, which was majorly online other than like a few little snippets. So the things we'll be covering is we'll first look at cell cytology and basics, then go to genetics and scientific investigation. All of these are super important as a base for everything you'll be covering in 3 and 4. So even though if they're not explicitly mentioned in the actual study design for 3 and 4, having an overview of these concepts will make it much better for you guys to understand anything that will be adding on to these concepts. So let's get started with the plasma membrane, and just a reminder to annotate the booklet as we go if you need to do that. So plasma membrane is obviously it's a phospholipid bilayer. So as you can see in the picture, it has two layers, and you need to understand how a single phospholipid looks as well. So if I was to annotate a single phospholipid, you have a head right here and you have a tail. So a head is usually hydrophobic. Head is hydrophilic and the tails are hydrophobic. And because of this arrangement, it is very selective in terms of what it allows to pass through and things like that. There's a fluid mosaic model as well, and it is known as fluid because the structure is very dynamic. It's constantly moving, and the phospholipids are also liquid at this state, at like room temperature. And the mosaic appearance is due to the carbohydrates and all the proteins embedded throughout the bilayer. Another essential component of our plasma membrane is cholesterol. So cholesterol is these little things that are studded throughout the membrane, and their main function is to provide stability and fluidity to the membrane. So like in hot temperatures, it prevents the membrane from being like too liquidy, and in cold temperatures, it prevents the membrane from being too rigid. So yeah, that's pretty much all for the plasma membrane. Now going through the protein secretory pathway, this is a study design dot point under area of study 1 in unit 3. So the main organelles that you need to be aware of is ribosomes, RUF-ER, the vesicles, Golgi apparatus, and the plasma membrane. So ribosomes are the site of translation where your amino acids will join together to produce polypeptide chains or proteins. So a really key detail to understand is that if the ribosomes are studded on the RUF-ER, then the protein that's produced on the ribosomes is made for export out of the cell. Whereas if you need to make a protein to use within the cell itself, the ribosomes would be used are free ribosomes. So they are not the ones studded on the RUF-ER. So every time a question asks you anything like ribosomes on the RUF-ER are used for what purpose, it's going to be to produce proteins for export out of the cell. So once ribosomes have made the protein, then my rough endoplasmic reticulum is going to manufacture them as well and do any processing and packaging that needs to be done. and it will be transported through the transport vesicles. And the transport vesicles' main function is to go from the rough ER to the Golgi apparatus. So yeah, I'll just go like this. Now, about the Golgi apparatus, its main function is to modify and package proteins for exocytosis. And something to note about the Golgi apparatus is that it has two sites. So the site in which the transport vesicle goes in is called the cis end, and the site in which the secretory vesicle is released is called the trans end. It's just a very minor detail to have a note of. And yeah, major function is modifying package. Secretory vesicles, their main function is to go from the Golgi apparatus to the plasma membrane for exocytosis. So once we reach our plasma membrane, exocytosis happens, which is pretty much when you have a vesicle, which has a very similar conformation to your plasma membrane, which allows it to merge with the membrane and expel its content out into the extracellular fluid. So there's a quick question here. I'll give you guys a few minutes to have a read over it. And yeah, let's answer that. You guys can just put your answers in the chat. Oh yeah, there's a close one. I don't think I can see what the answers are.
I can see we're still just waiting on a couple more people to participate. And then I'll close off the poll and show you the answers.
Sounds good.
Okay, so our results, most people have said A.
Yes, that is correct. The answer here is A, because it says that it's an intracellular protein, and intracellular proteins means that it's to use within the cell. So it is manufactured at free ribosomes. Well done. So now looking at photosynthesis. So pretty much photosynthesis, the main function of it is to transform light energy into chemical energy by forming sugar molecules such as glucose. It occurs in the chloroplast as well. And so specifically, like obviously animal cells don't have chloroplast, so it occurs in plant cells. And chlorophyll is an essential, I guess it's an essential substance that's needed for photosynthesis to occur. The reason is because if we look at the formula, it's carbon dioxide and water, including light and chlorophyll, producing glucose and oxygen. So the reason why light and chlorophyll are written on the arrows rather than part of reactants is because they are necessary for photosynthesis to happen, but they're not consumed or used as reactants within the actual reaction. Okay, so chlorophyll is super important in the light-dependent stage of photosynthesis. So there's two stages in photosynthesis, and the first stage is called the light-dependent stage, where the energy is provided through the light energy or any, so it's light. And in the light-independent stage, the energy, it happens in the absence of light. So any of the energy that is needed to carry out that one is provided through the ATP or the NADPH molecules, which are electron carriers that are produced from the light-dependent stage. So once we look at the inputs and the outputs, so obviously in our light-dependent phase, we're gonna input water, ADP pi and NADP plus. So ADP and pi and NADP plus, they are coenzymes. And coenzymes' main function is to assist the any of the enzymes that are involved in photosynthesis to carry out their reaction better. So ADP pi and NADP plus right now are unloaded versions because they're not carrying the hydrogen on them. So once the ADP and pi becomes your ATP, it becomes loaded, similar to an ADP plus, once it gets the hydrogen on it, it becomes an ADPH, and it becomes loaded as well. So we also produce O2, ATP, and an ADPH. These two are reactants as inputs in our light independent phase, whereas oxygen is a byproduct. So this is the common VCA question as well. I can't spell byproduct. Yeah, so it's like, what is a byproduct of light dependent stage? And the major byproduct that we have is oxygen, because it's not involved in the light independent stage. So the light dependent stage occurs in the grana of the chloroplast. So if we see the chloroplast diagram below, these little discs that we see are called thylakoids. And once the thylakoids stack up together, they're called a grana, or one of them is called a granum. Obviously, since they're green, it helps us identify that there's chlorophyll inside them. And the chlorophyll's main function is to absorb the light energy. Okay. And the light independent stage occurs in the stroma, which is like the cytoplasm of the chloroplast. So looking at the light independent stage, my inputs are ATP, NADPH, and carbon dioxide. So this is a new one that's being inputted. And pretty much the main function, it's also known as the Calvin cycle, and requires really high energy coenzymes to transform the carbon dioxide to glucose. So the high energy coenzymes refer to the ATP and NADPH that we see there. So light independent stage, once we input those, it will release the unloaded versions of ADP pi and NADP plus again, because the coenzymes, it's like a cycle. So even though enzymes are not used up in a reaction, coenzymes are. So coenzymes, if they are loaded, they'll become unloaded. So you need to do another reaction to make them loaded again. So it's like a cycle, like ADP plus pi goes to ATP, then ATP gets used up because it's the energy form, and then you need to recycle it back to ADP and pi. So that cycle continues. Yeah. And the major output that we need to care about is glucose, obviously. Something else to note is that in some reactions, you'll see that you actually need to, in the reaction, it may say there's 12 water on the reactant side plus 6 water on the product side. So in the actual exam, we only need to write a net equation, and that's why we can cancel out this water and cancel out the 12 to make that 6. Because ultimately what happens is that you need to input in 12 water and on 6 water come out after the light independent stage. But because if we look at the net equation, since you're inputting 12, you can minus the 6 that come out, and you'll be left with the 6 that actually went in the first place. So yeah, that's all pretty much photosynthesis is. And photosynthesis is part of Unit 3 area of study 2, and you are expected to know all the inputs and outputs of all the stages and where they occur as well. So now looking at cellular respiration. So cellular respiration refers to two things. It can refer to aerobic respiration and anaerobic respiration. So aerobic respiration pretty much is in presence of oxygen, whereas anaerobic is in the absence of oxygen. And it occurs in the mitochondria. So mitochondria is the powerhouse of the cell, but VCA doesn't. If you write mitochondria as the powerhouse of the cell in the exam, you would lose marks. So you would probably need to say it's like where energy is produced or where ATP is produced. And that pretty much highlights the main function of our cellular respiration, which is to produce ATP, to be used by the cells or any reactions that happen outside the cell as well. So in oxygen availability, the aerobic respiration occurs, which is more efficient than anaerobic because it breaks down the entire glucose molecule and it produces 36 to 38 ATP. So in the absence of oxygen, ethanol is produced in plants while lactate is produced in animals. So anaerobic respiration, since you don't have the oxygen present, it is incomplete and you're not going to get as much ATP out of it. So looking at the overall equations, your anaerobic respiration only produces 2 ATP. And the 2 ATP that actually produces is not in the second part of the anaerobic respiration process, but it's rather in the glycolysis stage. So that will make much more sense once we look at aerobic. So there's 3 phases in aerobic respiration. You have glycolysis, Krebs cycle, and electron transport chain. So glycolysis occurs in the cytoplasm, and Krebs cycle and electron transport chain occur in the mitochondria. So mitochondria is where the oxygen is mainly involved. So that means if you don't have oxygen present, glycolysis is still able to occur. Okay, and that's why in anaerobic respiration, glycolysis still occurs. It's just that Krebs cycle and electron transport chain aren't occurring. So aerobic respiration happens in mitochondria, whereas anaerobic happens in the cytoplasm. So pretty much looking at each step now, what glycolysis is, it's pretty much breaking down the glucose molecule, which has 6 carbons, into 2 pyruvate molecules, which is 3 carbon each. So what I mean by that is glycolysis. So glucose is C6H12O6 and pyruvate is C3. So obviously, since it's C6, you're going to be breaking it into 2 pyruvate molecules, and that's an easy way to remember the inputs and outputs of glycolysis. So yeah, Avika does want you to remember inputs and outputs of every single step and where they occur and what their ATP yield is. So glycolysis produces 2 ATP. Okay, Krebs cycle, on the other hand, it pretty much produces energy-rich loaded coenzymes for use in the electron transport chain. So it also produces 2 ATP. And lastly, electron transport chain, it's the most important one to produce the most ATP. And it transfers energy from all the electron suppliers to the loaded coenzymes to make ATP. So what I mean by the electron suppliers is that if we see in the diagram, alongside producing the ATP, we also have the molecules NADH, FADH2 that are also involved. And their main function is to act as electron transport or like electron carriers to go directly into the electron transport chain. So my inputs of glycolysis are glucose, NAD+, and ADP and pi. Again, the unloaded versions, and output will get the loaded versions such as NADH and ATP. Once we get the NADH and ATP, the NADH actually goes directly in the electron transport chain. It doesn't need to be an input in the Krebs cycle because the inputs in the Krebs cycle are also the unloaded versions, because the only thing where loaded is necessary is in the electron transport chain. So in the Krebs cycle, your inputs are pyruvate that you're producing from glycolysis. You're also adding in the NAD plus and FAD. Now, FAD is another coenzyme. So the unloaded version of FAD is FAD, obviously, and the loaded version is FADH2. And you're also adding ADP and pi. So outputs there will be CO2, NADH, and FADH2, the loaded versions, and ATP. So in the Krebs cycle, CO2 is my byproduct, just like how oxygen was my byproduct in photosynthesis. So once I produce those molecules, they will go to electron transport chain, where all of them act as inputs. So oxygen, NADH, and FADH2 are the inputs, and what you will get as an input and output is your H2O, NAD plus, and FAD, and ATP, obviously. So glycolysis, 2 ATP, Krebs cycle, 2 ATP, electron transport chain produces 32 to 34 ATP. So looking at it overall, cellular respiration, the aerobic version produces 36 to 38. There's just so it's 36 to 38 ATP overall. So now we also need to know where each of these stages occur. So as I mentioned, glycolysis happens in the cytosol. The Krebs cycle happens in the matrix of the mitochondria. So if I was to just drop a mitochondria right here, matrix of a mitochondria is like it's like the cytoplasm. So this is your matrix, this is your inner membrane, and this is your outer membrane. Okay, so inner membrane is also known as cristae. Cristae is where my electron transport chain happens. So it's really important to understand why it actually happens in the cristae as well. So it's because the cristae, cristae pretty much looks like this. So because it has this really folded appearance, it means it has a really high surface area. So the high surface area of the cristae would allow efficient absorption of the coenzymes, and this would increase my rate of ATP production. So high surface area increases rate of ATP production. And yeah, that's pretty much all for cellular respiration. And there's just 2 questions here, question 9 and 10. So let's take a moment to attempt these.
Okay, so most people have responded, and for question 9, the first question there, we've got the answer is A.
Yes, correct. So yeah, question 9 is A, really well done. That's the matrix. So do we know what reaction happens there? I guess you guys can just put it in the chat. What reaction happens at the matrix? Yes, correct. Krebs cycle. Well done. And question 10 now. Let's do that.
I'll relaunch the poll.
Thank you.
Okay, we've got some answers here again. I'll just end the poll. So there's a bit of a split. So some people are saying A and some are saying C.
Yeah. Okay. So option A is glucose enters glycolysis, and C says majority of the ATP is broken down and is produced in the cell. So the correct answer here is C. So the reason why the answer is C is because the structure labeled Y is actually my cristae or the inner membrane. So cristae or the inner membrane is where my electron transport chain happens. So it's where my majority of the ATP is produced. And as we know, electron transport chain produces 32 to 34 ATP. Glucose enters glycolysis. So glycolysis happens in the cytoplasm. And the illustration shown here is just the mitochondria. So cytoplasm isn't shown here. Okay, but well done. So there's just a 5-minute question timer for any questions that you may have. And it's like a 5-minute quick break. And so yeah, pull your questions in the Q&A or the chat. Yeah.
Okay, we've got a question, but I think that's more about like uni and stuff. So we can come back to that at the end. And I'm happy for you to just keep going on the content if you want.
Okay, sounds good. Yeah, so the next bit we'll be covering is all the genetics bit. So this is super important in once we look at Unit 4 as well as some bits in Unit 3. So the key terminology, also this is carrying on from Unit 2. So some of the words you will be familiar with. So starting off with gene, gene is a section of DNA that codes for a particular protein. And genome is the complete set of genes or genetic material that is present in a cell or an organism. So gene and genome are relevant once we look at what DNA is, as like once we actually start the Unit 3 content. And allele is just alternate forms of gene. Gene pool refers to the total genetic information that's present in a population. Genetic drift is random and unpredictable changes that impact allele frequencies due to chance events. So chance events are specifically, there's two types. There was one that was called the bottleneck effect, and there was one called the founder's effect. Even though it's not explicitly mentioned in the study design anymore, it was relevant in the last study design. So it is, I feel like there will be practice questions that you will do on it. So genetic drift is pretty much when you have unpredictable changes that you can't control. So bottleneck pretty much had this sort of shape. So bottleneck was something like this. So what happened was there would be a disaster or something like that that would make the allele frequencies. Allele frequencies is just like the proportions of all the alleles we have in our population. And so what would happen is that due to a chance event like a disaster or something, the allele frequencies would skew down. So you only had like a very few allele frequencies remaining. And the population will rebuild from the small skew. So obviously we would have a lot of genetic diversity that is lost in the process. And that was known as the bottleneck effect. The other example of genetic drift that we had was called the founder's effect. So founder's effect was like, you have a big population here with a lot of genetic diversity and a big allele frequency. And for some reason, like some of them, like for example, let's say these three, they decide to migrate to a new place. So now we see the allele frequencies in the new island are significantly lower than what we had on the parent island. And pretty much most genetic drifts would result in a reduced genetic diversity because like bottleneck, you had reduced right here and the population is rebuilding from there. And in the founder's effect, you had a reduced diversity right there and the population is trying to expand from the very few people that had migrated. Okay, so genetic drift is random and unpredictable changes that will impact the allele frequencies negatively most of the time as you will lose genetic diversity. Then you also have gene flow. Gene flow is pretty much the movement of individuals and their genetic material between populations. So gene flow is what allows similar species to exist within like different areas of the, like let's say in Australia, we would have like some birds in Victoria that we will also find in South Australia because gene flow is possible. So gene flow is just allowing their alleles and genetic makeup to be passed around. So if gene flow is halted, speciation may occur. So speciation is pretty much when you form the entire new species. So in order for speciation to occur, your gene flow must stop. So if gene flow stops, that means you don't have any transfer of genetic materials. That would mean your populations, like for example, I had population A and population B, and gene flow has stopped. For example, let's say due to an environmental barrier, like a mountain or something, then that means your populations are isolated, or what we would say is they are reproductively isolated because they're not able to reproduce with each other anymore. And if they don't reproduce, then that means they're not able to share their genetic material. So if they are reproductively isolated, and assuming they are also exposed to different selection pressures and things like that, they will evolve differently. So for example, let's say population A is in like humid conditions, population B is in very cold and snowy conditions. Due to the different selection pressures and different characteristics that are favored, the populations will be exposed to different selection pressures and will evolve differently. And if they evolve differently over time, they will become different species. So if they become different species, even if we take away the environmental barrier that we had here, nothing's going to happen between the population because now they are two species. So the way a species is defined is that if two populations are able to interbreed and produce fertile and viable offsprings. So if these population A and B were to interbreed, they wouldn't be able to produce viable, which means any offspring that are actually alive, and fertile, which means offsprings are able to reproduce themselves. So if they're not able to tick both those characteristics, that identifies that the populations are now separate species. And that's when we know speciation has happened. And the specific example that I just explained there was known as allopatric speciation. So allopatric speciation and allopatric speciation specifically refers to having an environmental barrier that halts gene flow. So genotype. So genotype refers to the entire genetic makeup of an organism. So it refers to the combination of alleles at a particular gene locus on a chromosome or something like that. And phenotype is genotype times the environment. So it is the observable characteristics resulting from a genotype's interaction with the environment. An example that I was taught about phenotype was about the traditional food binding in China. So pretty much in China, in olden days, they used to break the toes in order to make them appear something like this. So they would have no toes kind of thing. So the genotype would be like having the toes, and environmental effect would be having the cultural practice of binding the toes to look like flat. So once those two are mixed together, you would see the physical appearance of having foot bounded, which was like just what my fist is right now. Like you can't see any toes anymore. So yeah, phenotype is genotype times the environment. And there's a question on that now.
Okay, I think people have answered now. We've got a bit of a split between A and B.
Yeah, A and B, both really good options, and A is the distractor here. So A, the correct answer is B, is because it's pretty much saying if genome is referring to an organism, what refers to a population. So genome, as we know, is, once we go back here, genome, as we know, is the complete set of genes in one organism. So what refers to the entire genetic makeup in the entire population? So gene pool is what refers, because gene pool is, again, going back here, is the sum of the total genetic information in a population. So genome is the total information in an organism. Gene pool is the total information in a population. Okay, so yep, the answer here was B. So now we're gonna look at chromosomes and karyotypes. So pretty much chromosomes are, they are these structures that we see right here, and that's where our DNA is stored. So eukaryotic chromosomes, they're made of chromatin. And chromatin, what it is just really tightly condensed DNA, and it wrapped around histone proteins. So you have a nucleosome, and nucleosome is pretty much 4 histones and chromatin wrapped around them. And 4 histones mixed together are known as a nucleosome. And pretty much nucleosomes join together to form an entire chromosome. So yeah, sorry, it's 8 chromosomes, 8 histones, my bad. So yeah, section of DNA coiled around 8 histones is called a nucleosome. and they will join together to form chromosomes. So chromosomes typically have a linear structure in eukaryotes, while in prokaryotes, they have a circular structure. So if I was to draw a simple prokaryote, they would have their classic circular chromosome, but alongside having a circular chromosome, they also have these other circular rings, and those circular rings are known as plasmids. Now, plasmids are made entirely of DNA. They're very small DNA molecules that are self-replicating, and they're originally made by your DNA polymerase enzyme. And because they're self-replicating, we use them to do like gene cloning and things like that. Another really important property about plasmids is that they're able to pass across bacteria through conjugation. So I'm giving the specific example of bacteria here. So bacteria have the plasmids. So for example, if a particular bacteria was resistant to a particular antibiotic, it could pass on its antibiotic resistance to its neighboring bacteria through doing like a conjugation process with another bacteria next to it and passing down the plasmid. So plasmids are very small DNA molecules that have maximum of like 20 genes alongside having a circular chromosome. Okay, another really important thing to note between any eukaryotic DNA versus prokaryotic DNA is because prokaryotic DNA don't contain histones within their structure. So it's not as tightly wound, like even though they are tight, it's not like they're wound around any proteins. They are just like DNA. Okay. And the highly condensed nature of chromosomes is what allows them to be karyotyped. So chromosomes are effectively karyotyped during the metaphase of the cell cycle, because in metaphase, our chromosomes are the most condensed state. So they're most visible then. So looking at the picture, everyone has 22 autosomes. Autosomes are non-sex chromosomes, and we have 2 sex chromosomes. So males would have an X and a Y, and females would have 2 X's. Another thing I want to mention about DNA in general versus prokaryote versus eukaryote is that in eukaryotes, DNA has introns and exons, whereas prokaryotes don't have introns. So pretty much what introns is that you can remember it as introns are interruptions. So they are the non-coding regions of our DNA. And exons are excellent, so they are the coding regions of our DNA. So bacteria or any other prokaryotes don't have introns, which means when we look at like translation transcription in unit 3, like bacteria don't need to have a stage where you need to cut out the introns and join the exons together because they don't have introns to begin with. Okay, and that becomes really important when we look at gene cloning, because it's really important that when we add a human DNA into a prokaryotic plasmid or bacterial plasmid, that we don't add introns into it. Because if we add introns, the bacteria is just going to think that it's a coding region and actually translate it. We don't want that because it's non-coding. It's going to make a defective protein ultimately. Okay, so going back to our karyotypes, so pretty much a normal karyotype, as we can see, it will have 23 pairs of chromosomes, 22 autosomes, and 2 sex chromosomes. And now looking at mutations, so you can have 2 types of mutations overall. So you can have point mutations or block mutations. And under point mutations, you have missense, nonsense, silent, and addition and deletion. So missense mutations are when a nucleotide is, so pretty much point mutations are dealing with one nucleotide impacted. And block mutations are dealing with the entire chunk of chromosome being impacted. So point mutation is like, for example, a particular nucleotide is affected, and missense is where that nucleotide changes my amino acid. So obviously we know a triplet in DNA, which is 3 bases in DNA, code for 1 amino acid. So for example, if we see in this diagram right here, the TCC, it's being changed, the C is being changed. So here it's supposed to be T originally, but now it has a C. And once transcribed, it actually codes for an arginine amino acid instead of lysine. Okay, and that will change my amino acid. And if my amino acids change, this could potentially affect what kind of folding my protein has. So if you're producing a defective protein, you're gonna have a wrong type of folding. So for example, if protein needs like particular bonds to happen. So an amino acid called cysteine it produces disulfide bonds. So if you're replacing cysteine with another amino acid that doesn't produce disulfide bonds, then obviously your protein's not going to be folded in the correct way. And if the conformational shape is not correct in their tertiary structure, that means it's not going to be functioning as what it's supposed to function like. So missense mutation, amino acid changes. Nonsense mutation is pretty much when you have a premature stop codon. So what happens is that here again, TTC, you accidentally change it to ATC. So the T is changed to A, and that will, mRNA will transcribe that to a UAG, and UAG is a stop codon, which means that my polypeptide chain would be halted. And stop codon doesn't actually code for an amino acid, it codes for a release factor. So when translating a particular protein, once a ribosome reads the stop codon, it's actually going to release my polypeptide chain rather than adding a amino acid upon it. Okay, so stop codons would make like the defective proteins that are not complete. Silent mutations, they are the nicest type of mutations because they don't cause any change in any amino acid. So the property of DNA that becomes relevant here is called degeneracy or redundancy. So degeneracy. Yeah, pretty much what degeneracy or redundancy means is that multiple codons or multiple triplets can code for the same amino acid. So if we see here, AAG codes for lysine and AAA also codes for lysine. So that allows silent mutations to occur, okay, because the genetic code is degenerate and redundant. So yeah, silent mutation, even though you have a nucleotide change, it's not going to affect my amino acid. Now, alongside this, you also have two more point mutations, and these are jointly known as frame shift. So frame shift. The reason why they're called frame shift is because they quite literally shift the entire frame. So if you're adding a particular nucleotide, so for example, I have ATCCGA. Let's say I want to add another nucleotide right here. Let's say I'm adding another C. You can see even though this one remains the same, my next triplet is now CCG instead of being CGA. Okay, so after that particular point of mutation, all the amino acid are affected. Okay, because you're shifting the frame. You're adding something, and that's going to move everything one step down. So all your triplets and all your codons are affected. And the protein is going to be defective, or you're not going to produce the correct protein at all because all the amino acids are changed. Similarly, with deletion, let's say here, ignoring the C, if I was to remove the other C as well, you will still have ATC, but that means I have GA and something else like GAT. Okay, so you can see how everything is moved up a point because of a deletion mutation. Okay, so everything moves up and the protein is again defective because all the amino acid after the point of mutation have been affected. So those were all point mutations are when nucleotide is affected. On the other hand, block mutations is where a bigger chunk of the entire chromosome is affected. So you have 5 types there. You have inversion, duplication, translocation, deletion, and insertion. So inversion is pretty much what we see here. So you take a section of chromosome out, you flip it up, and you put it right back in. So it's just reversed. Okay, duplication is just like it sounds, it's duplicated. So you have double of that same thing. So a section of chromosome is copied, causing there to be 2 of the same sections. So in this case, if something is particularly overexpressed, like a particular protein is overexpressed or something like that, this can actually lead to cancer or any other health conditions. Then you also have a translocation. Pretty much what translocation is that you have 2 chromosomes, a section of one of the chromosomes cuts off and it goes and binds to another chromosome. So you can see the picture right here, like you can see the pink one's being cut off and it's being replaced with the yellow one that's also cut off. So one of the translocations could be like things are swapped around, or another type of translocation is that a little bit of a section of a chromosome is cut and it just goes and binds to another chromosome. Deletion is where a section of chromosome is completely lost, and insertion is a section of chromosome is added to another chromosome. So insertion kind of ties in with the translocation bit as well. So the most severe out of all of this would be deletion. The reason is because you're actually getting rid of genetic material. Like in all others, you're still having all the genetic material that your cell needs, like in your genome. Whereas in block, like deletion, you're completely getting rid of one of the chunks. So that means you're not going to be able to produce a particular protein at all. And that's dangerous to your body. So now looking at karyotype abnormalities. So you can have two types of abnormalities. You have aneuploidy and polyploidy. Aneuploidy is the most common one that's tested, and it's pretty much when you're missing one chromosome or possessing an extra of a one chromosome. So every time you're dealing with one chromosome, it's aneuploidy. If you're dealing with the entire set of chromosome, it's called polyploidy. So having one chromosome instead of normal 2 is called monosomy, and having 3 chromosomes instead of the normal 2 is called trisomy. Okay, and similarly, polyploidy is like, for example, if you have naturally everyone has 2 sets. If we have 3 sets, it's called triploidy. 4 sets, quadruploidy kind of thing. Okay, so the word is ploidy after. So yeah, polyploidy is the most common cause of human miscarriages, and it's not survivable. Whereas aneuploidy, there are some common ones that you can actually survive. So figure 10, we can see the trisomy 13, it's Patel syndrome. Trisomy 18 is Edwards syndrome. Trisomy 21 is Down syndrome. That's the most common one, and the one that most people are able to survive. Figure 13 is a monosomy of the sex chromosome. So instead of having like an XY or XX, you only have an X. So that's called the Turner syndrome. And what's unique about Turner syndrome is that whoever has Turner syndrome is always a female. Because in order to be a male, you must have a Y chromosome. So if you don't have a Y chromosome, then that means you can't be a male. So since Turner syndrome only have one X, they don't have a Y at all, they're going to be female. On the other hand, if you have a trisomy of the sex chromosome, which is when you have 2 X and also a Y, since you have the Y present, the person is going to be a male with some female characteristics attached. Okay, and lastly, figure 15, we can see triploidies. And triploidies are most common in plants, so like tomatoes, potatoes, like that. And like everything is tripled, as you can see in that picture there. So let's do another question.
Just waiting on a few more responses. Okay, here are the results. I think most people have said C.
Yes, well done. So Patel syndrome, very classic, 3 copies of chromosome 13 only. It's not a polyploidy because it's not the entire set of chromosomes being affected. So yeah, and that was a 2017 question, and question 29, which is expected to be hard because as you move on, like during the exam, the question gets harder. And yeah, any more questions that we have?
So we had one question come through in the direct chat before about your pathway to medicine, if you wanted to like talk about how you got into that at Monash.
Yeah, sure. So I applied through the undergraduate pathway, which is pretty much just doing the UCAT and applying as soon as I graduate. So Monash considers 1/3 UCAT, 1/3 ATAR, and 1/3 of the interview. And for me, they considered my ATAR and UCAT and offered me an interview spot looking at that. And through that, I got an interview offer. And ultimately what happens is that if you get an interview offer, they weigh the interview performance very highly compared to like your UCAT and your ATAR once you actually get the offer. And then all of them are combined, applicants are ranked, and yeah, and then like applicants are offered. I also have a bonded medical place, which means I don't have the normal cohort, which means pretty much bonded medical places where I have to sign a contract with the government saying like, if I study medicine after I graduate, I can help out for 3 years in areas which need doctors pretty much. So yeah, once I graduate, I'll need to go to any like rural areas or regional areas which have shortage of doctors and help out there. There's another scheme called the ERC along with the normal CSP place. ERC just extended rural cohort where you do your clinical years at Monash rurally instead of doing them at Metro. And it's a really good scheme as well. So if you want to maximize your chances of getting in medicine, apply to all of them.
Okay, yeah, hopefully that was really helpful for some people. Another question that I've had come through is how much of the exam is weighted on the year 11 content? Is it just pre-knowledge, or will there be questions directly about this stuff?
So there's not going to be questions explicitly about any year 11 content that's not showed up in year 12 as well. So for example, any genetic terminology that we looked at earlier was learned in year 11, but the thing is that you will relearn them or like add on to them in year 12 as well. So questions can be expected on those content. So any content from year 11 that is covered in year 12 is examinable, but year 11 content that's not covered in year 12 is not examinable. So anything like your Punnett squares and like pedigree charts is not examinable.
Okay, perfect. And last one before we wrap up with this question time was just about how you maximized your study time, I think. Yeah, study, and also how did you study for the UCAT?
Study for the UCAT. Well, I'll start off with studying for bio. So pretty much I did bio as an early subject in year 11 along with further maths as well. So that saved me a lot of time because I was able to focus on my 2 year 12 subjects in year 11. And because I did them early, it made it much easier for me to solely focus on them, because pretty much what year 11 is that you need to pass your unit 1 and 2 subjects. So unit 1 and 2 scores don't matter as much as unit 3 and 4 do. So unit 3 and 4 was my main priority in year 11. So that's what I focused on. And yeah, since I had bio and further, my major study technique was just doing questions. So I would like learn the content as it's taught in classes, or watch like controller videos regarding it, or like Amoeba Sisters on YouTube are great as well. And just do as many questions that I can get my hands on. So go-to was like checkpoints. Checkpoints are really good. And since the study design is changing, you make sure that like all the resources that you're using are relevant for 2022. So Vika has uploaded a new like practice, it's like sample exam on their website, but I would save that for end, because it's like kind of like the only 2022 exam you guys have since the study design did change. But start off by doing questions from like the really old. So Vika website has questions ranked like up to, I'm pretty sure I think it's like 2006 or something. You can obviously start looking at that and like do any question that's relevant. Obviously, the entire exam is not going to be relevant. So do the questions that are relevant. And yeah, that was my major study technique. I didn't personally make notes. I just used notes that I received from like tutoring or something, or that I received from my teachers. And I annotated notes rather than making my own, because I found that it saved my time. And spent most of my time doing questions, because Vika wants you to apply your knowledge rather than just reiterate the concepts. So doing questions and getting hands on as many questions as you can is the best way to apply, in my opinion. And as for the UCAT, so I started using, first of all, I started looking at the official website and doing some practice questions from there, because the official website has like small tutorials that are free to use, and they give out some like tips and tricks and explain what the UCAT actually is. So I found those really helpful. And there's also lots of practice questions on the UCAT consortium. And alongside that, I had commercial resources that I used as well from two companies. I don't think I'm allowed to share the names of the companies.
Nah, you can. Fine. Go ahead.
Oh, I can? Okay. So I used MetaTree and Metafy. So both were really good. Yeah, I recommend both of them. So Metafy was more actually mimics the actual exam, and MetaTree had harder, but like they had more questions than Metafy did. So having a combination. So both of them had questions alongside having mock exams and also having what's it called, resources to actually understand like tips and tricks to do the UCAT.
Okay, perfect. I think that we'll save the rest of the questions till the next break, but if you want to continue on with the content, I'm happy for you to do that.
Okay, sounds good. So now I want to move on to scientific investigation, which is something that you're going to do in all science subjects. So I did chem and bio, so I know about those two. I'm not fully sure about physics, but you definitely need it in chem and bio. And you can definitely expect exam questions on this. So looking at your key terminology, you need to understand what validity means. And this is where like the sticky stuff is, because like all the words sound the exact same: validity, reliability, reproducibility, repeatability, accuracy, and precision. Like these ones are super, super important to understand the difference between. So validity is pretty much the experiment measures what it sets out to investigate. Like it's like, does it fulfill its aim or not? So valid experiments, they will use one independent variable while everything else is controlled. And using only one independent variable, make sure that you don't have any extraneous variables that are impacting your results and things like that. And they're also not affected by experimental bias. So you have two types of validity as well. Internal validity is just measuring what they're supposed to. And external validity is like how much can you generalize your results? Like, are your results generalizable or not? Then reliability is pretty much an experiment which has high precision, repeatability, and reproducibility is called to be reliable. So you can rely on the results. The results are, see, like this is what I mean by the interchange. Like I was gonna say the results are valid, but like that's why everything is interlinked and it's really hard to pinpoint the exact differences. So something that helped me was like reliability is repeat. So every time you're repeating an experiment, you're increasing its reliability. So yeah, R and R, like repeating is reliability. Reproducibility, it's like how close are the results when the same variable is being measured using the same method and investigation, but under different conditions. So reproducibility and repeatability, they're both ways of determining precision. Okay, so reproducibility, how close results are when the same variable is tested, and repeatability is how close the results are in the exact same conditions, like successive measurements. So repeatability is what we tend to do in schools with experiments and stuff. It's more relevant. And yeah, so accuracy is how close an experimental value is close to its true value or like scientific value. And precision is like how close multiple measurements of the same investigations are to each other. So here's where the classic the dot analogy comes in. It's like, if I have a dartboard, like if you hit the bullseye, that's accurate. So like if you're constantly hitting the bullseye, like all of your darts are hitting the bullseye, then you're accurate and you're precise. But like, let's say if all your darts are constantly hitting this one point, that means you're precise, but you're not accurate because you're constantly hitting that one spot, but you're not hitting the bullseye, right? So next is my independent variable. So independent variable is the variable that is being changed. So on a graph, this is my x-axis variable. Independent is always going to be x-axis because it's the one being changed and it impacts your dependent variable. Dependent variable is what is being measured. So it is the one that's affected by the independent. So independent could be like, let's say, different concentrations of a particular enzyme, and dependent could be like rates of reaction. So if you're plotting that, you'll be like, as you're increasing the concentration of the enzyme, your rate of reaction is increasing. Like the graph would look something like that. So then you also have a controlled variable and the controlled experiment. So controlled variable is kept same throughout the experiment. You're not going to change it. And it's super important to have controlled variables because this makes sure that, again, only one independent variable is being tested, and this will ensure the experiment's validity. And this also makes sure that none of the extraneous variables are impacting the results as well. Then you also have a control experiment. So control experiment is usually conducted without the independent variable, and it sets a baseline. So pretty much the main function of the control experiment is to identify that any of the results that you're actually producing are because of the control variable or not. So the way a control could be carried out, again, I'm using the enzyme analogy. It's like if you are testing whether an enzyme, like how fast an enzyme breaks down a particular substrate or like any product, any reactants, then you would carry out a control variable without adding the enzyme at all and see what the rate of reaction would be if you didn't add any enzyme. So if the rate of reaction is 0, then we can assume that any changes in your rate of reaction can be attributed to your enzyme being present. So control experiment usually conducted without the independent variable, and it establishes a baseline for your experiment by establishing that the results that you're doing are because of the independent variable and nothing else, pretty much. So hopefully that made sense. So now looking at types of errors. So there's 3 main types. Again, this is also very examinable, especially if you look at the 2019 VCA exams last question. It was all about errors and like how to minimize the errors. So pretty much personal errors is random. It's like personal errors is like the experimenter is making the mistakes in like mathematical calculations and things like that. And they are reduced by repeating an experiment. So personal errors are really easily minimized. Then you also have systematic errors where you have issues with the experimental design itself. So if a particular experiment design, like method or something, it says to, It says to use a particular apparatus that is not accurate, then that would mean that you're constantly going to get the wrong results, no matter what you do. So even though your results will be precise, they're not going to be accurate. So they're consistently wrong values or consistently away from the expected values. In order to reduce them, you use the same piece of equipment when measuring data. You calibrate the equipment, or you choose an equipment with an appropriate scale, or measure the same substances with different pieces of the same equipment or like similar equipment, and then take an average of that. Then you also have random errors. So random errors are due to the statistical spread of results, which is normal despite repeating the experiment or like measurements several times. So pretty much you're never going to be able to get rid of random errors because they're always going to be present. So you can minimize them, but you can never get rid of them. So to reduce them, you could have the same person consistently take the measurements. So reading in pairs and coming to consensus as well, repeating the readings and taking an average. So all these are ways of minimizing random errors, but you can't eliminate them. So hypothesis, this is super common. Like this is like a very common exam question. Pretty much what hypothesis is going to be, it's like it predicts the relationship between your independent and dependent variables. And for hypothesis to be valid, it needs to be tentative, testable, and falsifiable. Pretty much what tentative means is that hypothesis is not certain, but attempted to explain based on theory. So hypothesis is your prediction. It's never going to be like 100% certain. But the only thing you can try and do is try and back up your any claims by making them as backed up by theory as you can. Testable means that you're going to be able to test the hypothesis by observations or investigations. Okay, and falsifiable is pretty much there is medians which would invalidate the hypothesis. Again, because a hypothesis is not 100% true all the time. So the most important thing is that how do we actually write the hypothesis? So VCA prefers if you use an if and then method. So usually you'll say like if, then you'll talk about what your independent variable is, and then you'll be like, then So if then, and you give a prediction involving what the dependent variable is going to be. So like if let me give an example of photosynthesis, because light is really important for photosynthesis. So if light was taken away during the experiment, then photosynthesis rate of reaction is going to drop, which we know because of theory, because light is an input in the light dependent reaction. So that's what when is referring to, like when change in independent variable occurs. So like if independent variable is this, then this is going to happen. And something else I personally like to use as well is that if you would want to add reasoning behind the answer. So I always did that in my practice exams and actual exam, is that like I would always add a because statement after I would say like if then, if the question was worth more than 1 or 2 marks. Most of the time the questions are 1 mark, but just in case if they are more, I would be safe and add a because statement. So like if light was taken away from a photosynthesis experiment, then photosynthesis rate of reaction is going to drop because light is an essential input in the light dependent phase of photosynthesis. Like very short and sweet, like only 1 sentence. It doesn't have to be too long. Another really important thing I wanted to mention that I haven't added in the slides is how to write a conclusion. So when writing a conclusion needs to be really short and succinct. And we actually marks on that. Like if a conclusion is really long, you actually risk losing marks. So you want to make sure your conclusion is very short, succinct, and to the point. So always mention whether your aim was fulfilled or not. Talk about whether your hypothesis was supported or not supported. Don't say proved or disproved, because you don't, I think the VCE examiner told me that like, you're not carrying out like a full-fledged experiment where you can disprove a hypothesis. So you would say hypothesis is not supported instead of saying hypothesis is disproved. So terminology is super important as well. So yeah, talk about aim, hypothesis, and summarize your results. And if the question asks like what you can do better in like future experiments, then maybe have 1 statement in that. But most of the times, conclusions are worth 3 marks. So 3 statements, 3 marks kind of thing. Just to be safe, add an extra statements, because like 4 statements, 3 mark is better just in case. Okay, and that's another question right there from 2018. Let's try that.
Sorry, we're just still waiting on a couple more answers. Okay, not everyone's answered, but I think we'll leave it there. The answer that most people have said is C.
Yep, the correct answer is C here. Okay, so the reason why it's C is because it's like group 3's measurements are precise, but they're not accurate. So if we see accurate measurements are, so like accurate measurements, we can identify them by looking at the other experiments. So for example, here, all of them are around like 18, 17 or stuff like that, right? The only thing is that group 3, their answers are consistently off. So their answers are consistently off, which indicates that even though their answers are not accurate, they are precise, because precision was again like hitting not the bullseye, but 1 spot on the dartboard consistently, like not like you're always missing the bullseye consistently. That's what precision is. Option A says group one's measurement are most accurate but least precise. So we can't eliminate, like we don't know, we're not fully sure about that because like we don't know what the experimental values are. Okay, group B's group two's measurements are accurate. B and option D can be ruled out because it says both accurate and precise. Well, we know they're not precise because the difference between 19 and 21 is more than 0.5. So 10, like usually it's like plus minus 0.5 is precise. But since it's way beyond that, it's not precise. Another really important thing I want to mention about scientific investigation as a whole is that like when you do scientific investigation at school, it's going to be typically when you carry out your own, experiment, things like that. But other than that, you don't tend to go into theory of like how you need to like structure your question or like how you need to structure your answers or like what any content you need to learn as well. So a lot of it is self-study. So refer back to this booklet for like all the definitions and things like that, but do a lot of practice like outside doing questions about scientific investigation because they show up on every exam and they are hard. Like the questions about scientific investigations are hard. So make sure you are able to attempt them because you're not going to learn them in like most schools don't teach them explicitly. But yeah, any questions before we do some more questions from the 2019 exam?
Yeah, we've got a couple questions that have come through. One is just asking, how did you balance your time in year 12? Is there like a secret to it, or should you just do what you think is right?
Well, balancing time online was easier because like my school didn't run Zoom lessons half the time. So it was a lot of it was self-study. So I would just do obviously the content we learn in class, but alongside that, I only had 4 subjects to do in year 12. But because I had 4 subjects to do, it made it much easier for me to focus on the UCAT exam as well because UCAT prep needs to be done. Like you need to consider UCAT as like the whole subject itself, and like you need to commit to it, and you need to prepare for it as you would for any other subject. But like UCAT exam is in July. So you only have like 6 months in the year to prepare for it. So you need to treat it as one of the subjects. And like most schools will also have like study hall or like any study free periods, and make sure you utilize those effectively. Not gonna lie, I didn't. I just wasted my study session because first of all, like since we were not at school, the only time we were at school and would get study sessions at school, I wouldn't want to study because we weren't at school for most of the time. So I just want to be like, I want to talk to people instead. So if you guys, I'm hoping that you guys are actually back on campus this year. So if study classes are on campus, make sure you utilize them effectively. And if your school does have a quiet study space, use that. And really important to not just focus on like studying in year 12, because like year 12 is so much more than that. I wish we still had our events, but all of them were canceled. But like just enjoy it as much as you can. Like even though we didn't have anything, like year 12 was still like my favorite year because like everyone just connected better to each other. Like we all were able to help out each other. So talk to a lot of people and make friends because like have fun because it's like the final year of high school anyway. So there's no point in wasting all of it in just studying. So have a good balance by setting like priority lists for subjects and like don't leave exam prep to the last minute. Be consistent. So do everything consistently from the very start.
Okay, perfect. And let me just look. The last question, would you recommend having a tutor for biology, or do you think it's okay to just do it by yourself?
I personally had a tutor for bio, And I found it really helpful because so what happens in classes is that sometimes teachers would rush through particular content or like not explain it as well as you would want it to be. So even though you have extra help such as a Drolo and things like that, I still recommend a tutor because it's like your own personal point of contact. Like if there's a topic you're not understanding, your teacher's not going to spend an entire lesson explaining it to you unless you have like really helpful teachers, which I didn't. So a tutor was super helpful for me because I could direct all my questions to them and even go through any like practice questions that I didn't understand like fully or something like that. So just working through questions and content with them would allow me to like focus on the issues that I'm facing in that particular subject rather than going at the same pace as what the class is doing as well. So yeah.
Okay, perfect. And this isn't in the chat, but for me, are you available as a tutor at the moment, or have you already filled up your spots for 2022?
I am not available just yet because maybe I could take on like 1 or 2 more students, but I'm not fully sure. But my availability on KIS is unavailable for now, though. So I could make that available. So if you have the link to my KIS thing, if I'm not available, I could still like link you up with some really amazing tutors from KIS who have also scored like really high.
Yeah, so what I'm just going to do is to everyone on the thing, I'm going to send a link. This is our marketplace. So KIS Find a Tutor. You can find a VCE tutor for literally any subject that you are struggling with. And they're all super helpful. So yeah, just reach out to that link and click request a tutor. You get a free half-hour trial with each of them. So use that, see if you like them. And yeah, if you haven't already and you're doing chemistry or methods, we do have 2 more of these events. Chemistry is tomorrow and methods is on Saturday, same time as today. So if you found this helpful, do that. But now for the remaining like 15-ish minutes, we're just going to go through a past paper.
Yeah, I'll just share that. Okay, can you guys see my screen? So this was the first 2 questions on the 2019 exam, which is the exam that I did. And it's pretty much about protein secretory pathway and protein transport. Protein transport has been actually taken out of the study design. So, no, sorry, not protein transport, like transport across the membrane has been taken out of the study design. So just attempt question 2.
Okay, so we haven't got any responses coming through just yet. Did you want to kind of give them a hint, maybe talk them through the first question?
Yeah, okay. So question 1 was pretty much like what's the type of transport that we can see? And I feel like question 1 will help you kind of with 2. Actually, not really. But so let me just go to question 2 directly because question 1 was not too relevant here. So question 2 is like, what organelle synthesizes proteins from their monomers? So first of all, let's see, what are the monomers of proteins? The monomers of proteins are amino acids. So it's asking, at what organelle do amino acids join together to form proteins? It's a step in translation. So I'm not sure if you guys have looked into transcription translation just yet, but it's like, where does translation happen sort of thing. Translation is how proteins are produced. So what organelle synthesizes proteins?
Oh, sorry. I think the poll I have up is for the first question. So I'll just end that poll and relaunch for question 2.
Okay, no worries.
So this is for question 2 now, guys. Put in your answers for that. Okay, yeah, those answers are coming through. We've got a bit of a split between A and B.
Oh, okay, A and B. So Golgi apparatus and ribosomes. So Golgi apparatus' the answer here is actually ribosomes, because Golgi apparatus' main function is to modify and package proteins. So at Golgi apparatus, you will have things like glycosylation and phosphorylation, which is not something that you need when you're actually making the proteins from their monomers. So monomers, again, were amino acids. And once amino acids are joined together by condensation polymerization, they form a polypeptide chain. And the polypeptide chain is what the protein actually is. And that process happens at the ribosomes, which is the site of translation and protein synthesis. Yeah. So let me go to another. Okay, this is a bit of questions 7 till 9 are all about experimental design. So have a read of it and let's see if we can do question. Let's do question 7 first.
Okay, so I've relaunched the poll for question 7. Just while you guys are working, I would like to remind you that this is the actual VCE exam, so it just shows how relevant all of this content is, and yeah.
And for question 7 as well, like you can see the structure and all the answers, like all of them follow the if and then format. So it's super important that in your exams, you also adapt to the same method of writing it.
Okay, the responses have mostly come out as D.
D, yes, that is correct. So if the temperature increases, the bacteria will grow more quickly. And we can see that because -10 is pretty much 0, 15 it increases, 25 it increases further. And the reasoning behind this is because as we increase the temperature, the rate of reaction of our enzymes actually increases because they have more kinetic energy. And if they have more kinetic energy, they're actually able to move around much quicker, and they're able to carry out the reaction much quicker. But if the reaction temperature goes beyond their optimal, the enzymes would denature. So denaturation is where they'll stop working. Okay, so let's look at question 8 now.
Again, just while you're working, for those of you who are interested in the UCAT, this is not through KIS, but Suzen is actually going to be running a UCAT sort of like workshop in February. So if you're interested in that, I'm just gonna put in the chat to everyone. This is Suzen's number. She said she was happy for me to give it out. Oh, Suzen, I spelled that wrong. I'm sorry. About UCAT, message her on there, and she'll get you set up with a link to that webinar. My other announcement, just super quickly whilst you're doing this, is if you want to give us feedback about this event, if anything worked super well or if something wasn't quite up to your satisfaction, which we hope everything was, but we really want your honest feedback. So please let us know at this link. It's just a general feedback form about the event. And yeah, we'd love to hear from you if you have anything to say. Okay, again, most of the results have come out as D.
Yes, correct. So Reasoning is because independent variable is the one that you're changing. Dependent variable is the one you're measuring. So independent here will be your temperatures because you are exposing them to 3 different temperatures as it's written here. And what we're testing is the percentage of nutrient agar covered by the bacteria, which was D. Yeah, well done. And last question is question 9. It's a very buzzwordy question. If you read the word reliability, what's another R word that you should pick?
Okay, I think everyone has responded now and they've said A.
Yes, correct. So reliability always works with repeat. If you want to increase reliability, you will increase the repeats. And yeah, that's pretty much all the questions I had. That's like, as I was mentioning, like if I just open VCA Bio exam, like on the actual VCA website, alongside having these exams, you'll also have access to NHT exams. So NHT exams are also made by VCA. And the NHT exams are pretty much for people who are sitting the VCA exams for any of like the subjects that are available here, like in the Northern Hemisphere. So yeah, like year 2018 and 2017 relevant exams. But yeah, on the actual website, it's up to 2002. So like I would like look at the exams early and just like do any like questions relevant, or like even ask your teacher for any questions that they may have, or like any topic tests or practice exams or SACs.
Okay, awesome. Thank you everyone for being so engaged today and responding to our polls and asking questions for Susan. It makes it such a much better experience to be presenting to an active audience. So thank you for that. If you have any questions, please feel free to reach out to us at KIS. All of our contact information is on our website. And again, we've got more workshops coming up tomorrow and on Saturday. So look into those if you haven't already. And please, thank you to Susan. Thank you for all of your hard work putting the booklet together in the PowerPoint, and then of course presenting today. We really appreciate it. And yeah, if you want to give us feedback or tutor at KIS, all the information is in the chat. So I'll leave the event open for another couple of minutes, another couple of seconds for you to copy that if you're interested. And yeah, thank you all for coming today. And thank you, Susan.
Thank you so much.
Wrap up now. As I said, last thing, sorry, the recording will be available on our online courses. Just send us an email. We have a record of all the people who bought tickets, and we'll be able to give you free access to that online course if you want to review the recording. Okay, awesome. That's all from me. Thanks. Bye, guys.
Bye.